his own rights. Let's have a big and warm applause for Professor Charles Rice. Distinguished guests, esteemed faculty members, students, ladies and gentlemen, good afternoon. It is a profound honor to welcome you to Tzu Chi University. Today, we are privileged to host a visionary lecture by Nobel Laureate Professor Charles Rice. We are delighted to have you to hear for this extraordinary academic exchange. Before we proceed, allow me to introduce our guests of honor. Please join me in welcoming Professor Charles Rice, Professor of the University and our keynote speaker, Mr. Uwe Molavez, Chairman of the International Peace Foundation, Dr. Jia Hongdao, Vice Superintendent, National Taiwan University Hospital. Mr. Wen-Chang, Minister on Home Assignment, Institute of Diplomacy and International Affairs, Ministry of Foreign Affairs. Mr. Hualien, CEO of Buddhist Tzu Chi Charity Foundation. Mr. Ching, CEO of Buddhist Tzu Chi Medical Foundation. Dr. Huang, CEO of Tzu Chi Education Foundation. Dr. Chen, Superintendent of Taipei Tzu Chi Hospital. Dr. Liao, President of Tzu Chi University. A warm welcome to you all. To formally commence today's proceeding, let us invite Mr. Hualien, CEO of Buddhist Tzu Chi Charity Foundation, to deliver the opening remarks. Mr. Yan.
Distinguished Nobel Laureate Dr. Charles Rice, Chairman of International Peace Foundation, Mr. Uwe Molavez, CEO of Tzu Chi Medical Foundation, Dr. Liao, and President of Tzu Chi University, Dr. Liao, Superintendent of Taipei Tzu Chi Hospital, Dr. Chen, and all the guests, esteemed scholars, students, good afternoon. It is my great honor and privilege to welcome all of you to this very special occasion. Today, we are deeply honored to host Dr. Charles Rice, recipient of the 2020 Nobel Prize in Physiology or Medicine, whose groundbreaking work on hepatitis C virus has transformed global health and saved countless lives. Dr. Rice's dedication to scientific discovery stands as a shining example of how knowledge perseverance, and collaboration can bring hope to humanity. On behalf of all of us here, I extend the warmest welcome and deepest respect to you, Dr. Rice. I would like to also to express our sincere appreciation to Mr. Wu for his visionary leadership in launching the Taiwan Bridges event series. This meaningful platform connects Nobel laureates and distinguished scholars with Taiwan, fostering dialogue, collaboration, and exchange of knowledge across borders. Through this platform, bridges are not only built between nations, but also between minds and generations. I would like to recall a meaningful milestone in this journey. In June 2024, Mr. Wu, together with Mr. Raymond Song, the founder and chairman of Liao, actually who is the largest sponsor of this program, and President Chen Wen-Chang of National Taiwan University and myself had the honor of visiting President Liao. During that meeting, we shared a common vision to bring Nobel laureates to Taiwan to foster deeper academic cooperation and cultural exchanges. The Taiwan Bridges event series later on evolved into Taiwan Bridges Initiative, led by Academia Sinica, National Taiwan University, and the International Peace Foundation. From November 2025 to May 2026, 31 Nobel winners will deliver lectures at over 10 universities and research institutions across Taiwan. Today's gathering is a beautiful realization of that shared vision. Once again, I extend my deepest gratitude to Mr. Wu and all partners who have made this meaningful exchange possible. And most importantly, we thank Dr. Charles Rice for sharing his wisdom, experience, and inspiration with us today. Thank you.
To invite the host, Dr. Liao, the president of Tzu Chi University, to address the assembly. Please, President Liao.
Good afternoon, everyone. Welcome to Tzu Chi University. On behalf of Tzu Chi University, I would like to thank all of you for your participation. Also, I would like to thank International Peace Foundation for your support, and also the support from all Tzu Chi 4 missions, especially the support from Tzu Chi Charity Foundation. This afternoon, I'm looking forward to an inspiring scientific interaction. So, students, faculties, please get your questions ready and raise your hand. For high school students, if you ask good questions, and I will write a recommendation later for your application to the university. Okay? Enjoy the scientific fest. And thank you again for your coming. And thank you, Charlie, for coming all the way to inspire us. Thank you.
Now, to introduce our keynote speaker, it is my pleasure to invite Dr. Chen, Superintendent of Taipei Tzu Chi Hospital, to the podium to offer his introductory remarks and introduce Professor Rice.
Dear Professor Rice and dear friends, it's my great honor to be here to say a few words before Rice's speech. I'm 4 years younger than Professor Rice. 1988 to 1990, I worked at Michael Lai's lab at USC. By that time, Professor Rice already earned his PhD degree at Caltech and moved to Washington University. In 1989, one day Science published a paper published by Michael Horton and his fellow Zhu Kunling. Dr. Zhu was graduated from National Taiwan University. We read that paper, and from that time on, many biologists focused on this topic. By that time, we were busy with coronavirus and the Delta virus at Michael's lab. And Professor Rice was invited to join the study of HCV. Later on, you may learn a lot from his speech. But the most important contribution from Professor Rice was by that time, replication of HCV is very, very difficult. But Professor Rice and his colleagues found that over the 3' end of the non-translating region, there are very, very important sequence of poly-A or poly-UC. And later, they also found that 5' end also have poly-UC tail or poly-UC sequence. They are very, very important for HCV replication. Later, Professor Rice proved, after inoculation, the HCV transcript into the liver of chimpanzee, proved that the viral genome we found is really the HCV. The most important contribution Professor Rice enabled us to have weapon to combat with the HCV. Dr. Rice developed the DAA agent. That safe and effective agent can cure all the HCV patients. So right now, we are so lucky that whenever HCV patient came to you, you can, yeah, I'm a good doctor, I can cure you. So right now, nowadays in Taiwan, almost all HCV patients are cured. So all of this was Professor Rice's contribution to the human. I admire the most is not the intelligence or talent in science, but is his determination and perseverance, because he encounters so many difficulties and failures and frustrations during the whole process of his research. So Professor Rice is not a hero in combating the virus, but also a role model we should follow. So it's my great honor here. Thank you so much to come to Hualien, to Taiwan. Thank you so much. You enable us. Thank you.
And ladies and gentlemen, let's welcome Professor Charles Rice to the stage for his lecture.
I think I have to be unmuted. I am. Okay. I don't think so. Thank you, Bill. Well, it's a great pleasure and honor to be here and to meet so many Taiwanese scientists. And I have to say that it was also quite an experience to meet the board of high school students that were here. So it's always wonderful to see youthful enthusiasm shining through, and I really thought that at the outset, I'm going to give kind of a personal journey on this subject of hepatitis C. And it really has been a long road. And despite the fact that there have really been significant breakthroughs in treatment, I think this is something that we see in other challenges in public health. We still have a ways to go. So if we get back into sort of the early history of hepatitis, and this is really more for some of the younger folks, but it was already recognized that centuries ago that there were these outbreaks of hepatitis leading to sort of jaundice, sort of yellow skin color due to liver damage. And really sort of coming to the 1900s, sort of Max Tyler, and I'll talk a little bit more about this later, developed a vaccine for yellow fever that they ended up stabilizing with human serum. And this vaccine actually caused hepatitis, but it wasn't due to this live attenuated yellow fever. And it was recognized in both people and in animals that transfer of serum could elicit these episodes of hepatitis, that there was a link between hepatitis and human serum. So it was recognized in the 1940s that there were really sort of two kinds of agents. One that was probably transmitted via the fecal oral route, responsible for these outbreaks sort of during wars and so on. And then ones that were sort of a serum transfer, like I just described the yellow fever case. So in 1965, and I think Baruch Bloomberg, I heard, has actually been a speaker in this series. Baruch sort of discovered hepatitis B virus in human serum. And then in 1973, Steve Feinstein, Dr. now particularly, sort of discovered hepatitis A virus. And this was one that was transmitted by the fecal oral route. And in the early to mid 1970s, was sort of the ability to sort of diagnose hepatitis A and hepatitis B viruses, it became clear that there was something else going on in the transfusion population. And this was discovered by Harvey Alter and at the NIH and Fred Prince. And it was really quite common. So that 10% of the transfused patients would develop hepatitis. It was often a persistent infection. It could be transmitted to chimpanzees, our closest relative. And it was probably a small envelope virus. That's because this agent could be inactivated with organic solvents. So, today you would think, okay, it's going to be no problem to sort of figure out, what the virus is that's responsible for this. But it turned out that took sort of nearly 15 years to accomplish. And as we've already heard, in 1989, there was a seminal paper published by Michael Houghton and his colleagues sort of reporting the sort of discovery of this non-A non-G agent, which was echoing a little bit there. Maybe we could turn this down a little bit. Anyway, it was an expression screening protocol that they used to identify a cDNA clone of this non-A non-G virus. And this occurred after years of attempts at trial. So this paper appeared in Science, and basically the things that caught my attention here were that it was an RNA virus, likely the positive strand RNA virus, and it was probably similar, at least in terms of the genome size, to members of the Coviviridae or the Coviviridae. So those were two virus families that I actually been working on as a graduate student as a postdoc. So this was 1989. I'd been at Washington University for about 2 years at that point. And here was this sort of new virus that was related to something that we've been studying. And we have been studying yellow fever, which fortunately is not a problem for the current people. But it's the prototype member of the Kobe family. And I already mentioned this remarkable live attenuated strain that Max Tyler derived by basically pathogen a wild isolated yellow fever in minced up tissues of chicken embryos and mouse embryos. And this vaccine is kind of the ideal vaccine that one would like to have. It's basically one shot, we believe, lifelong protection from, as far as we know, sort of all wild strains of yellow fever. So really quite amazing. And for us in the laboratory, this was a convenient virus to work on because it grew in cell culture. And actually we could be working with the virus that was the vaccine strain. So, you know, at least it wasn't something that was going to kill you if you made a mistake. And Max Tyler won a Nobel Prize for that in 1951. And we determined the genome sequence of that in 1985. And then in 1989, we had, you know, one of the things that RNA virus people like to do is to be able to introduce mutations and understand, you know, sort of what the consequences are for the virus. And that's a problem with RNA because you don't really have the toolbox of modifying RNA in a site-specific manner, at least in those days. And so the way that this was accomplished was actually to take these RNA viral genomes, transcribe them into complementary DNA, and then basically use that complementary DNA, which can be propagated as a plasmid in bacteria like E. coli, as a template to transcribe basically a mimic of the genome RNA of the virus. And since positive strand RNA viruses have basically all of the information that they need in the incoming genome to initiate a complete replication cycle, you can take that synthetic RNA and introduce it into a cell, and then basically see what the mutation is that you were studying. So we were very interested in doing this with yellow fever because we have the vaccine strain, and we also have the sequence of the virulent parental strain. And being able to take these to a DNA intermediate will allow us to understand more about the basis of why this strain is attenuated and why it is a good vaccine strain. So we were really excited about this. We had published the initial yellow fever sequence in Science, which was a big journal and still is. And so we sent this infectious clone paper on the vaccine strain to Science. And they sent it out for review. We got the reviews back. And they were pretty good. We had some more work to do. We did that and sent it back to Science. And then they said, well, yeah, we did sort of answer these critiques, but we just don't think this is the general interest. So at about that time, there was a new journal, a new biologist that was going to become the next, you know, Science, Nature, you know, sort of cell-like journal. And they were interested in this paper, so we sent it to them, got the review back. It got accepted in the new biologist, and then this journal went extinct in about 2 years. So I don't think it was the yellow fever paper that its demise. But one of the things that we had put forth in that paper was this statement here that may allow yellow fever 17B to be used as a carrier for immunologically important epitopes from other disease agents. The idea being that maybe we could re-engineer the yellow fever vaccine strain as a live attenuated immunogen for other infectious diseases. And in fact, that turned out to be the case. But it was really, you know, I was really on the edge of working on hepatitis C, I have to say. And this is just one of these things in life that happens that can change your life. And I got a call from Steve Feinstein. I already mentioned that he was one of the co-discoverers of hepatitis A virus. And working at NIH, and was working on an ion creation. And Steve was at the U.S. and he was interested in developing vaccines. And he saw this paper that we published in the biologist, and so we called up for that. I had never met him. And he said, yes, well, what about making a chimeric virus with basically HCV because it was a huge global problem. And basically the estimates of this have varied over the years between 200 million chronically infected people to over 100 million. So a lot of people chronically infected with this virus, not uniformly across the globe. And the amazing thing about this little tiny RNA virus is that when people get infected with it, most people can't kill the virus. So it's not like, when people resolving a cold virus infection, we have symptoms and then they go away. But this establishes chronic infection in the liver. It's an exclusively liver virus. And basically, over time, that can progress to inflammation in the liver, cirrhosis, and end-stage liver disease, and liver cancer. So it made a lot of sense to really try and sort of do something about this. And with the discovery of the virus, he was relatively quick where the blood supply was being done, so that the chances of getting this from a transfusion are essentially zero. High-risk groups, this is a sort of blood-transmitted virus, so in groups of people who inject drugs, this is a common virus. We still don't have a vaccine. I'll say a little bit about that at the end. And we'd really like to come up with a successful way of eliminating this chronic infection where, we really didn't have sort of a lot of cases where this had ever been possible in the past in infectious disease, so to improve treatment. Early treatment for hepatitis C was actually using a cytokine called interferon, and this is an antiviral state. But it comes with a whole collection of side effects that people were not keen on, because often this chronic infection with hepatitis C is relatively subclinical. And that's one of the problems with it. People sometimes don't sort of get diagnosed with hepatitis C until they've already sort of initiated or developed cancer. So it's a problem. So I guess as a virologist in public health, we like to introduce this virus. So the problems with tackling hepatitis C is that this is like the worst possible virus that you could imagine to work on if you were a young investigator. We had spent a lot of time setting up systems where we could study alpha viruses and flaviviruses. But in the case of hepatitis C, even in the non-A, non-B sort of era, people have been unable to propagate this virus in cell culture. And the only way of assaying the virus was the chimpanzee model, which is sort of really not a model that you can do a lot of fundamental experiments with. So the early studies were really taking this sequence, and this is a schematic of what the viral genome looked like at the time. This sort of element of the 5' negative genome is basically an RNA element that's important for initiating protein translation. Then it had this basically long open reading frame of over 3,000 amino acids. And I was very lucky at the time that Barashka, who joined the lab, he was the first sort of technician at our hire, originating from Iran, but he'd actually been a student at Washington University. And we decided that despite the fact that this was like the worst possible viral system to study, we started working on it along with many other groups around the world. And basically discovered and mapped that this long polyprotein and 3,000 amino acids got cleaved up into sort of 10 different proteins. And if you looked at the sequence of these particular proteins, they could actually be proteins that were essential to the virus, like a viral protease that's responsible for making some of the cleavages that separate these guys. And also an enzyme, basically, that the virus is really running depending on RNA. So there was basically structural data on some of these enzymatic assays. This is the serine protease helicase and that's 3,4A and 5B, which is the RNA-dependent RNA. So the problem was that we had really no way of really proving that these were essential for virus replication, because we didn't have a system where we could say make mutants of these that would be expected to abolish the activity and see that they inhibited virus replication. So again, for hepatitis C, as we had done for sort of yellow fever, we started to try and construct an infectious form, the cDNA form of this virus, in which we could derive this form of RNA. And so well-behaved viruses, I think many of you can appreciate, a virus infects that permissive cell, and then you make a lot more virus. So that wasn't possible with hepatitis C. If you extract RNA from these positive viruses, you can transfect it into a permissive cell and get virus. So that's another way to go. You can also reverse transcribe the RNA, put it in a plasmid, as I mentioned, for yellow fever, and then sort of use that either to directly transfect cells and launch the virus, or you can use it as a template to basically make synthetic genome RNA equivalents and transfect those into cells and keep them together. And the question was, what source of hepatitis C material are you going to use? And it turned out that Harvey Alter at the NIH had collected some heat-based serums from a patient called patient H, or Mr. Hutchison, that had been titrated in the chimpanzee model. It was highly infectious, so there was basically not a lot of infected sort of viruses in this preparation. And so this, as I said, was tighter than chimpanzees, precious in limited quantities. And so we basically, in collaboration with Steve Feinstein, started to put together a CDNA clone of this patient which isolated basically. And so we did that, and we tried transfecting those RNA transcripts into cell culture, believing that even though we didn't have a cell culture system that was permissive for infectious with the virus, if we bypassed that infection step by just transfecting in RNA, we still might see evidence of RNA replication and amplification. But that didn't happen. So the only other alternative was actually to take this RNA and inject it into the liver of the chimpanzee. Now, most of you have not worked with RNA, although obviously it's a pretty important molecule for our life. But it's actually very unstable and very easy to get sort of degraded. So you could imagine that just injecting sort of naked RNA, if it wasn't protected by being a virus particle into the liver of an animal, wouldn't have a high probability of success. However, there has been an example of that from Bob Marcel at NIH on hepatitis A virus, where they've been able to make synthetic hepatitis A virus RNA and inject it into the liver of a marmoset and get successful launch of hepatitis A. So we went ahead and, again, Steve Feinstein, our collaborator at the FDA, was essential for doing these chimpanzee experiments, and we struck out. So that was disappointing because these chimpanzee experiments were not cheap. So we're going to infect a chimpanzee in those days with hepatitis C. You actually had to endow the animal, and it cost about $86,000 per animal. So, at least for me as a young assistant professor, if we hadn't had a collaboration with Steve at the FDA, we would have never been able to do these experiments. So I was fortunate to have an immigrant from Russian Sasha Poligoff from here. And we really got worried about the 3' MTR, the sort of non-translated region of the genome of the 3'N. And that's because the original chiron sequence had suggested that there was a poly-A 3' track, which was typically a cellular messenger RNAs. And in the case of some work from Asia, primarily in Japan, sequencing the 1B strain, actually they found this poly-U, the polychromium track of the 3'N. But we actually knew from our work on yellow fever, and really this is one of the things that I kind of wanted to get across today, and that is that even my mother, when I said we were working on yellow fever, she said, why are you working on yellow fever? Don't we have a vaccine for that? And it was really that experience, I think, of working on yellow fever that put us in a position where we could begin to study a new virus like hepatitis C. And that was very important. We knew that yellow fever did not terminate with the homocolumnar track. It instead terminated with this very stable standard structure. Sasha went on to sort of identify that, and we also was identified in Carol by Kumatata Shimotono in Japan. And basically it was a sequence of about 98 bases shown here. but have an internal polynucleotide track. And it turned out, if you look at different HCV isolates, this is a very heterogeneous virus, that this was probably the most highly conserved RNA element in the viral genome. So it kind of made sense if you tried to initiate virus infection with an RNA that didn't have this, it wasn't going to work. So, was this the missing piece? We were pretty confident it was. We went ahead and sort of made a new version of our H-strain cDNA, made RNA, and it failed again. We felt it was sort of proper. So this was, again, sort of really disappointing. And I have to point out that this was 1995, so we're already 5-6 years after the discovery of the virus, and we haven't really cracked this challenge. So, what could be wrong? Inefficient RNA delivery, part of the genome missing. Maybe hepatitis C wasn't like other positive strand viruses. It needed some other modification or another protein or something before it initiated infection. And in those days, the enzymes that we had for sort of amplifying and cloning were not as high fidelity as they are today. So there were probably mistakes during cDNA cloning. As I mentioned, this virus is very heterogeneous. And so what we ended up doing, and what Sasha put together, was basically a cDNA clone where every nucleotide in that clone reflected sort of a common nucleotide in the virus population, a consensus sequence of references to try and filter out changes that might be mistakes. So we made those constructs, sent them off to Steve, and this was really kind of an aha moment in the sense that both of the chimpanzees that were transfected with this RNA became virulent, and that's shown here in blue. It accumulated over time. We've never seen anything like this in other attempts. And in red, you can actually see a marker of liver damage from damaged cells. That's when the immune response kicks in and recognizes that something is going on in the liver and begins to attack these virus-infected cells. So this was a eureka moment, I guess, if you will. We ended up publishing this in 1997, and that work was followed shortly by parallel studies at the NIH for Jens Luckman and Bob Snow. So this really proved that we could actually artificially construct this genome, put it into an animal, and it caused the same kind of infections that we saw in sort of the original patient-age bacteria in the chimpanzee model. So we finally knew what a functional HCV sequence was, and this really sort of proved that it's sufficient to cause disease. So you would think, having overcome this barrier, that it would be easy to establish a system. We had an RNA that we knew worked. And it had been validated in the chimpanzee model. It was tried by us and many other people in various cell culture models, and it failed miserably to replicate. So again, we had sort of no way forward to actually develop a cell culture system, which is going to be important for not only fundamental virus biology, but also for developing drugs. So the next breakthrough came from Ralph Bartenschlager and the Volker Roma at the University of Mainz in Germany. And they were working with a different isolate of HCV, a genotype 1B isolate called CON1. And they basically made this, what we call a subgenomic replicon. So they chopped out the portion of the open region thing that would encode the structural proteins in the virus and replaced that with a drug selective marker. And then basically drove the rest of the HCV genome, presumably expressing the RNA replication machinery that this replicon would need in order to amplify. And when they transfected this into a human hepatoma cell line, HLA7 cells, and selected with, basically the neomycin analog E418, they had very infrequent colonies that grew up that had persistently replicating HCV RNA. So this was the first hint of a cell-based system where HCV RNA amplification could occur. The problem was that it was really inefficient, and only about 1 in a million transfected cells sort of allowed this to happen. And it turned out that if you sequence these replicon-containing colonies, that the sequence that you've recovered was not the one that you started. And in fact, they had acquired mutations that allowed them to replicate in these HLA7 cells. And some of these single amino acid substitutions in the replicon could increase the efficiency of the system by more than 50,000-fold. So really quite amazing. And then we could also cure some of these hepatoma cells that harbor replicons with interferon or other treatments and recover cell substrates that we've been working missing. So we finally have, really an efficient replication system to begin to study this virus. So a lot of the work in hepatitis C after 1999 really sort of used this system to understand more about replication. It also really became the driver of drug discovery. As I'll tell you later on, the sort of targets that emerged as sort of good antiviral targets for inhibiting hepatitis C were all present in the discovery of the replicon. So you would think that now that we have these adaptive mutations, it would be simple to actually get a complete system for hepatitis C. So we and others sort of engineer these adaptive mutations back into sort of full-length and modified clones. And then these replicated, they expressed the proteins, but there was no virus. So again, another sort of roadblock encountered. Here we're out, sort of more than 10 years, sort of after the discovery of this virus. So the next breakthrough in this field came from some work in Japan by Takashi. And Takashi was studying patient material from these very rare cases of severe acute hepatitis from hepatitis C, thinking that maybe the sort of more aggressive disease course was indicative of a virus that might have enhanced ability to replicate in cell culture. And so he began studying one that was called Japanese pulmonary hepatitis 1, JFH1. And we were all actually quite excited about this isolate because when subgenomic replicons were made using JFH1, they were able to sort of replicate efficiently in these hepatoma cultures without adaptive mutation. And we were wondering if these adaptive mutations that allowed persistent replication were somehow deleterious for later steps in virus formation and release. So it turned out that that was true, and so JFH basically could be used to infect cells. And virus was produced. This was infectious in chimpanzees or humanized, liver-humanized mouse models. And so we finally had, after all these years, and this is 15 years, sort of after the discovery of this virus, a complete cell culture system. Something that we take for granted in terms of these other viruses. So this is a movie that we were very happy with. Sort of most biologists are happy when they see a virus sort of kill cells or form plaques or foci. This is a live cell imaging movie of hepatitis C infection in hepatoma cells where viral proteins are appearing on the left, and then there's a reporter in the cells on the right, where if it's cleaved by the viral serine protease, the reporter relocates in the nucleus and shows up as a nuclear signal. So I think this really ushered in a golden age of research on hepatitis C. People could begin to understand more about the virus life cycle, virus-host interactions, began to study innate and adaptive immune responses, and look for other HCV-related viruses in culture. I won't have time to tell you about this today, but one of the areas that has been frustrating hepatitis C has been the inability to really understand more about the pathogenesis of virus infection. A lot of that has to do with not being able to actually do experiments that are really rigorous in the chimpanzee model. We can't use those anymore in the States. And we have collaborated with others to identify hepatitis C-like viruses in nature. One of those actually turns out to be a virus that was sort of running around the streets of New York in Norway rats. And that has turned out to be a very decent model. So it's one of the best studied RNA viruses. And let me just say a little bit about treatment. I'm going to leave out sort of a lot. really, sort of between these early days of being able to work with the virus and treatment. But this virus, in some respects, has properties that make it more amenable to cure. So it's not like HIV, the genome, it replicates in the cytoplasm, it doesn't integrate in the genome. We knew from the interferon treatment days that successful treatment does lead to a cure, and the virus is gone, and that if you eliminate the virus, it lowers the risk of liver disease. So it turns out that the work in sort of biotech and pharma sort of gave rise to molecules that are sort of directed against these three vital targets, the serine protease that I mentioned before, the RNA-dependent RNA polymerase, and then the NS5A, another non-structural protein that's involved in hepatitis C RNA replication and virus assembly and release. And these things are really quite remarkable. So these cocktails that are typically just sort of two of these antiviral compounds are capable of eliminating the virus in 2 to 3 months of treatment, sort of 1 pill a day. And as I mentioned before, hepatitis C is a very heterogeneous virus. And one of the numbers that really sort of gets me sort of thinking about how lucky we are that these treatments are so good is that in a single chronically infected individual, there are an estimated 10 to the 12th variants being produced per day. And so you can imagine there are all kinds of single amino acid substitutions and 2 amino acid substitutions. How are you going to actually come up with a compound that doesn't show antiviral resistance? But these combinations really do that. So it's been quite a road, and I've inflicted a lot of sort of molecular virology on you guys, and I hope that's not too bad. But this started out as a mystery virus back in the 70s with a 5% cure rate with systemic interferon that made people quite old. In 1989, it was identified, and that led to sort of diagnostics to clean up the blood supply. In 2011, the first of these direct-acting antiviral agents was combined with interferon therapy. That increased the cure rate to about 75%. That was a very sort of toxic regimen. And then in 2014, 2015, we refined it out of the era of interferon and into basically orally highly effective drugs. So in terms of some of the, you would think, sort of with an advance like this, that it would be like, flipping a switch. We've got a cure for this virus, so let's just identify everybody, treat them, and we're done. And that turned out to not be so easy. And you guys have done a fantastic job of this in Taiwan, but that's not true throughout the world. And identifying who's infected, we really need, sort of a public health system that, checks people. Implementation, actually getting somebody from diagnosis to, taking their drugs and actually sort of resolving the virus. There are basically some barriers there. This treatment started off as being really expensive, and it still is in some places, but here in Taiwan, if you are diagnosed with hepatitis C, you have access to these drugs, you get treated, and you're done. That's not true everywhere. With hepatitis C and HIV co-infection, which is common in this virus, these were patients that were very difficult to treat. With these new regimens, you really see that that's not a problem. Virus resistance hasn't been too much of a problem, and we still don't have a vaccine for hepatitis C, which we feel would be important to sort of add to our content. So to admit sort of a difference here, this graph basically shows a ranking for most cases of acute HCV in the world with various countries. In 2015, which is when these direct-acting antiviral cocktails started getting used, and 2020, 5 years later. Now that basically on a global scale, decreased the incidence from 64 million to around 57 million. With 5 more years, I think we're down into 48 million people. But if you look at the United States, our sort of ranking in most cases has actually gone up. And that's due, I think, to the opioid sort of epidemic that we're having in the U.S. with more acute infections. And if you look at Egypt, or if you look at Taiwan, basically, look at the ranking of Egypt is dropped 12 places. And that's because in Egypt, the prevalence of HCV was about 10% of the population. So they knew that they had a ticking timeline. And this was created by a medical intervention for cysticercosis that ended up getting a large fraction of the Egyptian population chronically infected with HCV. But they managed to negotiate a deal with one of the pharma companies to be able to manufacture these antiviral drugs in Egypt and also supply them at a price that the public health system in Egypt can afford. And now they've managed to treat more than 95% of their population successfully for HCV. So there are initiatives sort of by the WHO to try and eliminate viral hepatitis in that, hepatitis C in particular. And in Taiwan, this was sort of what you were supposed to achieve by 2030. And here in Taiwan, you actually sort of met that goal 5 years ahead of schedule in 2025. So that's how sort of things should be done. We've got the drugs, we've got basically national health insurance here, screening programs, decentralized care. Gets the job done. So, I congratulate you in the sense that this is really, sort of a role model for, how we should be going around the world. And I guess one of the things that I'm not going to have time to talk about it, but some of the new areas that are enabled by these fantastic drugs that we have for treating hepatitis C we could consider basically a one-shot cure. These compounds are so potent that it should be possible to formulate them so that if somebody is diagnosed with hepatitis C, you could actually inject them in sort of a long-acting regimen, and they'd be cured in 2 to 3 months. So I think that would actually, for some of the populations that we're dealing with, then we wouldn't be losing patients to care. So I think that's something that people are considering. In the case of solid organ transplantation, as many of you know, there are a shortage of organs for people that need them in order to live. And now this treatment of acute HCV infection is so efficient that we can actually transplant organs from HCV-positive donors into HCV-negative recipients. And they become infected with hepatitis B, but as far as I've seen, they are all successfully treated and resolve the virus after transplant. And then the other thing that people have been sort of discussing is a possible way of accelerating vaccine development for hepatitis B. And vaccine development is very difficult because we have a lot of different platforms that we can consider for making vaccines. But it's very difficult to decide which of those is actually going to work in people. And you can imagine that developing a vaccine for hepatitis C would be tough in terms of a field trial because you would be, sort of targeting a population with high risk of acquiring hepatitis C. These would be potentially sort of young people who inject drugs. And so these trials are very, very difficult. So people are actually considering the possibility of establishing a human challenge paradigm where people could be tested for immunogenicity in terms of the sort of vaccine candidates and then actually challenged with the virus to see if it prevents chronic infection, because that's really all you need to do. So that's some of the things that people are considering together as well. So I hope I haven't bored you too much, probably terribly, but one thing that I want to sort of emphasize is that when you have somebody like me doing a talk and sort of being a co-laureate of this Nobel Prize, it kind of ignores the contribution of all of the other people that have been involved in this endeavor. And it really, I think in the hepatitis C community, even researchers were competing against each other to, sort of advance breakthrough. I think it was actually a very friendly environment, where, sort of, if somebody made an advance, I think it was celebrated by everybody. And so it was actually a great research community. And it was a very long road, from this sort of discovery of something going on in the mid-1970s to sort of where we are today. And we still need to go. And I wanted to just read this quote, from Lewis Thomas, who's gone now, but he was just sort of a great thinker of medicine and I think in biology. And I just love this quote. I think it's really important because one of the things that you notice in terms of biology is that there are, a lot of challenges out there. We don't necessarily know where they're coming from in terms of pandemic preparedness. But I think we have to, cast a wide net, even for things that are not obvious causes of human disease, to be ready for the next pandemic. And so what Lewis Thomas says, in the course of scientific progress, things tend to pop up. So if something pops up and you don't expect it, that's kind of what you're hoping for, if you're a scientist, because it's not what you expected, so maybe you're on to something new. And have their achievements have their origins in moments of surprise in the laboratory that are planned for and anticipated and predicted. No committee convenes to survey the future prospects of biomedical research, possibly a guess at the things that lay ahead. And I think we're seeing that now in many aspects of biology. No branch of government could have laid out detailed plans for the scientific needs of the future beyond asserting that what the country needed was more fundamental knowledge about the human form and function, about the agents and influences responsible for disease, and in a certain sense, about nature itself. Scientific research works, and it's the only way to get the underlying mechanisms of disease and the only way to learn what to do about it. And I think that's as true today as it was in the mid-60s. So, in terms of Taiwan, I'm going to sort of say a few things about hepatitis B, because this was a huge problem in Taiwan. And I think that the scientists and clinicians in Taiwan really made a significant contribution to getting this under control. And I'm just kind of, without getting into details, just highlighting a few of the things, but I think the sort of universal vaccine program here and the outcome of that was actually the first proof that vaccination could prevent cancer. And if you add up all of the sort of cancer deaths that have been prevented by the HBV vaccine, I think it probably dwarfs all of the sort of cancer successes in other areas. And the same thing is going to be true for hepatitis B as well. So I think that was really important. We saw that also for papillomavirus as well, and cervical cancer. Proof that vaccination can prevent cancer. And then establishing that high serum DNA, the strongest predictor of disease. So that says that if you are chronically infected with hepatitis B, suppressing the virus is important, and probably doing it sooner rather than later is a good idea. Lots of great molecular biology here. Epidemiological clustering for hepatitis C sort of allowed sort of very effective strategies, and as I said, well ahead of the 2030 measure. And again, integrating treatment into national insurance so that everybody can get it. And lots of molecular biology and animal models. Now, the reason I bring this up, because I think Taiwan has pretty much got hepatitis B under control, is that in the rest of the world, that's not the case. And we still have about 300 million people that are chronically infected with hepatitis B. And, we have 1 million infections a year, almost a million deaths due to viral hepatitis-related cancer. And in most cases, the treatments for hepatitis B are not cured. So I just put this slide up as a plea for some of the incredible scientists here in Taiwan that probably know more about hepatitis B than just about anybody on the planet to consider maybe part-time continuing to sort of work on this global challenge. So final thoughts, I think one's career is often determined by chance encounters, and that was certainly true with me. I sort of mentioned one with sort of a call from Steve Feinstein, but there are a lot of other sort of binary decisions. I think that when we decided to actually start working on hepatitis C, the advice that I got from the more senior members of the department at WashU was you were crazy to do this. And I thought maybe we were, but it turned out okay. Who you encounter makes a difference. That is certainly true. So I think the sort of mentoring, sort of look around for people that you can learn from that can support you, because this is really, if you go into science and medicine, this is a long career. You're going to sort of need advice and support throughout that time. Persistence can pay off, and sometimes things are just not that easy. I think in the case of hepatitis C, I tried to paint a picture as to how many sort of roadblocks that we and others encountered before we got to the finish line. Sometimes you just have to suck it up and go in the next day and sort of try the next experiment. And it's not easy, but if you really believe that you are going to make a difference, you can if you really sort of persist. And this is also sort of related, fundamental curiosity in research. In my case, that was sort of driven by our work on yellow fever. There was very little known about this family of viruses. We knew that they were important for pathogens, mainly Japanese encephalitis, so it made sense to be studying them. But we were allowed to actually work on yellow fever. As I said, there's already a vaccine strain because there was some flexibility in the system that we could just sort of work on things. And there's another example of a Taiwanese scientist, Liao. We got interested in coronaviruses and worked on mouse hepatitis virus. My goodness, why are you studying the mouse hepatitis virus? And we now know that the contributions of those people that worked on coronaviruses in those old days were absolutely essential to sort of understanding what we were dealing with when SARS, MERS, and COVID-19 sort of emerged. So really important. I think that the tools that you young scientists have today are simply amazing. I think combining that with AI and machine learning, which is obviously a strength here in Taiwan, has the promise of really giving us some real insights, some gold nuggets from these complex data sets that set us on the right track. And we really need to sort of harness this and encourage our young scientists that this is something that they should consider doing. So this is my last slide. Never give up. And I just want to thank all of you for coming. And it's really been a pleasure to visit Taiwan again and meet all those new faces. Thank you.
Please return to your seat for a short rest for a moment.
Okay. I'll sneak off.
Before we move to our Q&A session, we would like to take this opportunity to express our sincere gratitude. Our deep thanks to Tzu Chi Foundation and the dedicated team at Tzu Chi University for their hard work and commitment in making this event a success. Let's give them a big applause. We are also truly grateful to Professor Rice and our honored guests, Mr. Uwe, for traveling such a long distance and joining us today, despite the jet lag. And please join me in a warm round of applause for them. Thank you very much. And we will now begin our Q&A session. We are only begin our Q&A session. We are honored to have Dr. Tsing to moderate this session.
Professor Rice, and we don't usually have such a big crowd at the lecture, so this is very impressive at our school. And I heard that Dr. Rice enjoys interacting with students. So let's keep the atmosphere lively and take this opportunity to ask questions. So now it's time to open the floor for questions. Just a gentle reminder, if the audience on the 5th floor would like to ask any questions, please raise your hand and our staff will give you a microphone.
Thank you, everyone. Hello, good afternoon. My name is Ravi. I am an international student, PhD student at National TAIWAN University, and my current research involves using artificial intelligence for healthcare technologies. So, as mentioned in your presentation as well, and I researched it before, like my country, Pakistan, is ranked second most affected by the HCV virus after China, given the fact that China has like 1.4 billion population, but we just have like 250 million, but we are still the second most affected nation by that. Given that AI, artificial intelligence, and automatic technologies are evolving rapidly these days, so how do you see AI and deep learning contributing to the viral disease detection or early diagnosis in the next decade? In your opinion, what are the biggest risks of relying on those automatic systems in medical diagnosis? And finally, what advice would you give to young researchers who feel overwhelmed by the interdisciplinary research? So, to summarize, my question was, how we can use AI and deep learning automatic technologies for viral disease detection or early diagnosis. And second one was, what are the biggest risks on relying on those techniques? And finally, your advice to the young researchers. Thank you.
So this was an AI question. It was a little tough to sort of hear from the sort of echoes, but you were worried about sort of what are the benefits of AI and sort of what are the risks with respect to virology?
Yeah, my question was, can we rely on these techniques or on automatic technologies for early detection? Because when we do it manually, it takes time. So if we diagnose those diseases early, so maybe that will help us, prevent the disease early and cure it at time, and maybe overcome and reduce the cases.
Well, I'm not exactly sure what the question is. I don't know if any of the other folks up there can figure it out. But I mean, I think that in terms of AI and sort of virology, I think first of all, as I sort of alluded to at the end, I think we are in an era where we're able to sort of generate increasingly complex data sets. Sort of whether or not that's, sort of with single cell sequencing or some of these ways that you can actually sort of trace development. And I think, sort of AI is very important for trying to sort of tease out sort of the nuggets that are going to be important with those kinds of data sets. I mean, I do worry a little bit in the sense that, sort of what you get out of AI is, a function of what AI is learning from. And so, it's very important to still have a source of generating, sort of good old reliable data that sort of feeds into these models. So I think it's just another, sort of fantastic tool that we have, but you can't always believe it either, right? I mean, I think when you sort of ask AI something, and maybe this just reflects my ineptitude at using the technology, but if it's something that you really know a lot about, then you can often sort of see things that just aren't quite right in the output. So I kind of worry about, people accepting this as the truth without really sort of doing their own, rigorous fact-checking before deciding on the next experiment. So I don't know if this is at all related to what you were asking, but
it was perfectly related. You answered both the question, the first two parts, like how can it help and what are the risks? Like you mentioned that we cannot design a billion model. The last question was, what will be your advice to the young researchers who are overwhelmed by the interdisciplinary research?
we'll sort of discuss it at the
We will save time for other questions, okay? All right, thank you. Other questions?
Thanks for your excellent talk. After so many years, I'm still surprised to see the result that AcRNA is the consensus sequence from an infected patient can be infectious in chimpanzee. My question is that in that specific experiment, what is the predominant sequence or consensus sequence in chimpanzee after several rounds of infection? After infection, after you inoculate the RNA into a chimpanzee, after several days, what's the predominant sequence of the ACE in chimpanzee?
So your question was sort of about the vaccine? No, sorry. Virus evolution. Well, I mean, in terms of hepatitis C, as I sort of mentioned at the end, the number of variants in a single chronically infected individual is huge. And there are 6 major genotypes. And I mean, we're actually very lucky that these drugs have been sort of optimized to work. Now, in terms of evolution of the virus,
that's why I'm curious. Suppose a virus to cause cross-species transmission, it requires mutation, or the consensus sequence from an infected patient can be infectious.
Well, I mean, in terms of cross-species transmission, that's an interesting point. We still have no idea where hepatitis C came from. So we don't know, sort of what the sort of interspecies transmission was. I mean, with next generation sequencing, many different related viruses to hepatitis C have been found in nature, but none of them really all that close. So it wouldn't, there's no evidence that there's, a non-human primate in Africa that, for example, has something more closely related to human hepatitis C. So we really still have no idea where it came from. In terms of cross-species transmission, in terms of hepatitis C, we tried for many years to actually develop a mouse model for hepatitis C, and trying to sort of define, what are the human factors that the virus really requires, and could we engineer a mouse that expressed those? And we did that, and we actually developed an HCV permissive mouse, but it was basically resolved very quickly and caused no disease. And that's why, we were sort of excited about this rat virus that was isolated in New York, which can infect laboratory mice, and actually you can elicit a chronic infection, and it leads with high penetrance to liver cancer in that model. So, but you know, those, I think Norway rats and laboratory mice are actually phylogenetically fairly closely related. So maybe we were just lucky that the rat virus worked in mice.
Thank you. And any questions from high school students?
Okay, I see you.
Okay. Microphone.
Hello, here. Thank you so much, Professor Isaac. I'm actually not a high school student, but I just thought I had a quick comment. I just wanted to thank you so much for that presentation. When I learned about some virology back in cell line genetics 20 years ago, back then there was no cure, per se, for Hsing. I felt really hopeful after today's presentation that we are now at the age where there's a possibility, if not a direction, where it could be one day no longer a death sentence of some sort. Because back in the day, anybody who had Hep C would just be kept at bay and that's it, right? Like there was no actual cure. So you would try medication after medication. So thank you so much for that. The one small question would be, is there any sort of preventative measures that people could consider as well to go along once they've been diagnosed? Are there things that they could do on the side to be able to help themselves better take the medication on their journey back to health? Thank you.
Okay. I'm sorry I'm having trouble understanding these right from the, but could you state your question slowly and maybe I can, or you could just come up here on the stage and tell me.
Okay, well, if you don't mind, I'll just come up.
This is the audience participation. Come on up.
Okay. So my question was, are there any preventative or sort of lifestyle, I guess, accompaniments that people can also do on their journey back to health? Because once they've been diagnosed, they probably will be put on the medication so that it can help them recover from maybe HCV. So I was just wondering if there's anything that people should sort of do to help themselves through the entire process.
Well, that's a loaded question, and you know, sort of how do you know, it has to do with sort of this implementation, you know, sort of pipeline. And I think people are making some real efforts to sort of come up with mechanisms of sort of enhancing from A to Z. In other words, you know, getting people in to sort of being diagnosed with hepatitis C, and then sort of guiding them through the process of, you know, getting their prescription and, you know, taking. That's one reason why I kind of like, at least in terms of treatment, you know, one shot and you're cured. And then the other thing that's true in hepatitis C, I think, as well, in individuals that have more advanced liver disease, we can still very effectively rid of the virus. But in many of those people, you know, basically carcinogenesis has already happened. So they're still at risk of developing liver cancer. That's one reason why we're excited about this model, in the sense that maybe we can understand more about, you know, what the biomarkers are in hepatitis C patients that where cancer has been initiated and are still at risk. And that's also true of hepatitis B as well.
Yeah, that's part of the reason why I thought, oh, well, are there any sort of follow-up things that people can do as they're recovering? Because it's a little bit of a process. I mean, I know ideally it would be lovely to just have a one-shot cure and then you're 100% healed, but I know there's a little bit of a tail.
I don't know. After you sort of live through the era, sort of where people would get these injections of interferon, for 6 months or 12 months, or some of them even like, 2 years. Sort of 1 pill a day for 2 to 3 months isn't, sort of too much to ask.
Yeah, that's probably the best case scenario.
All right, thank you.
Thank you so much.
Okay.
Okay. I know there's I to inform the question from high school students. And once again, a small reminder, when you ask the questions, please ask clear and slowly, and also make a short statement. Okay, thank you. One question at a time. Okay, one question at a time.
Hello, Dr. Rice. I'm a high school student from Tainan Tzu Chi Senior High School. And your discovery of the hepatitis C virus took years of persistence, especially during the times when the virus simply wouldn't replicate in the lab. As a high school student, we often face obstacles and setups in our studies or life that make us want to give up. And when you face a long series of failures, how do you maintain a positive mindset and find strength to keep moving forward without losing hope?
Oh, okay. The question was, when things go wrong, how do you sort of motivate yourself to, sort of keep going? I don't know. I mean, I think at some point you are, I don't know, I've never had a problem saying, oh, this didn't work, what should I do next? I have no idea. So I think sort of thinking about, sort of what you've done and just sort of being engaged about sort of the next step. I have to say, in this hepatitis C journey, sort of with so many of these things that sort of didn't work without a great deal more effort, there were two things that were important. I mean, one is that you still need to have something going on in the lab where things are happening. So it isn't like we stopped working on tocoviruses and flaviviruses, so we still continue sort of working in those areas. But I think also, you just kind of get stubborn and you say, this has got to work, and I'm gonna get it to work, or die trying. And so maybe I just got a sort of a stubborn streak. Because, we had succeeded in doing this for other viruses. We should be able to do it for hepatitis C. It's just that we didn't have tools that were easy. I mean, with SARS-CoV-2, it's not a big deal. Somebody, sneezes, you sort of take a swab, put it in culture media, maybe filter that before you dump it on substrate cells, and then the next day you've got a virus cell. A little different than hepatitis C.
President Liao?
Thank you for inspiring talk. I am a high school student and I really appreciate the insight you shared today. And I would like to ask that during the research process, especially in the early stage when outcomes are still uncertain, are there any particular signs or criteria that help you decide whether a problem is worth dedicating several years to.
Okay, that was sort of a current question about sort of the direction of sort of science in the United States. Do I have that right? Well, it's a tough time. I have to say there's a lot of uncertainty, and I think in the case of infectious disease research, we're kind of in the bullseye of the sort of new administration. I really thought that, sort of after the worst of COVID-19 and seeing on a global scale and a national scale what a pandemic virus like that could do to the world, that there would be strong support for having a sustained effort in virology and microbiology, vaccine development, efforts to really sort of optimize our ability to respond in the future. And in fact, that's not turned out to be the case. At least in the United States, we are facing some significant uncertainties. And it's very difficult, I think, right now for people that say they were, sort of wanted to be on an academic track, but what does the future hold? And I think the most important thing to do now is to really sort of maintain some momentum, because I think that this is really an area that is not going to go away. We can't just sort of give up. So I think it's, we'll try and fight this as we can sort of in the United States. But sort of when science gets, politicized like this, it's a real problem. When facts don't matter, it's a real problem. So I think right now, just sort of, this is an opportunity for, science in other countries to shine. Because I think we're going to be in a little bit of a downside here on the U.S. side. And I hope that will be transient and reversible. But it is a concern. So I don't know if that gets at your question. It's not like, don't worry about it. I mean, I think, people are worried about it. But, if you really have a passion in the infectious disease space, one of the advantages that we have in sort of infectious disease research is that we often know what the infectious agent is. And that's a huge advantage in terms of being able to study it and come up with solutions. So, I mean, at least in terms of improving human health, I think it's still a very attractive area to think about working in.
Thank you.
My question goes to Deputy Superintendent Liao. I think Taiwan is about to claim that we have eliminated the hepatitis C. Would you please tell us what is the key policy or strategy that we did right? And is Taiwan's experience can be shared to other countries?
Okay, no problem. I think I should give the speech issue about the vaccination in Taiwan to President Liu. I think this is a very important issue. As Professor Rice mentioned, we need to make a difference. I think since viral hepatitis is quite endemic in Taiwan, so in the very beginning, I think my mentor's mentor, Professor Song, and also my mentor, Professor Chen, they identified that the most liver disease in Taiwanese people are caused by hepatitis virus because the Professor Bloomberg identified hepatitis B surface antigen, or it is called Australian at that time. So they do an epidemiological study and identify that for the general populations, the HPV SNG positive rate may be around 20%. And for patients with chronic liver disease, liver cirrhosis, and liver cancer, the liver cancer can be up to 80%. So suggesting that HPV infection is a major etiology of chronic liver disease in Taiwan. So I think with this, the background information, the government acknowledged that the viral hepatitis, especially HPV at that time, is the major threat to the health, the people's health in Taiwan. So from the government level, they have a hepatitis control committee, and then in the cabinet, they have different the policy makers, and also the scientists, the clinicians, and also the biotech people. They organize a platform trying to understand. And at the time, I think there are many studies suggesting the vertical transmissions, the mother-to-child transmission is an important transmission route. I think the Dr. made a tremendous effort at the time. So if we want to interrupt the transmission, I think the best way is to have the hepatitis B vaccinations. So at the time, I think in the early 1980s, the hepatitis B vaccinations was developed by the United States company, but at the time there is no clinical trial in the world. Maybe in some HIV people in the United States, but in other countries there are no such clinical trials. Therefore, the government and also Professor Song, Professor Chen, they developed the hepatitis B vaccination program. So as Professor Rice mentioned, so it was because the earlier clinical study showed its effectiveness maybe up to 90%. So since 1984, the government decided to have universal vaccinations. And now, after 4 decades of vaccination program, nowadays the HPV SNG care rate has already dropped from 20% now to less than 1% in those who are vaccinated. So this is a successful story of hepatitis B. And for hepatitis C, as the 3 Nobel Laureates, Dr. Michael Houghton, Dr. Harvey Walter, and also Professor Rice, they made the different contributions to the discovery and also the elimination of hepatitis C. As in the Michael Houghton's and the TAIWAN company, they identified the HCV genome. And Harvey Walter, through the blood bank study, know the hepatitis C virus is a major etiology of post-transfusion hepatitis in the United States, I think other countries as well. And Professor Rice, the infectious HCV clone and in vitro cell culture system for the drug discovery, that make the HCV cure possible. So let's make a difference. So because of the success of HPV program, so I think the government grabbed this opportunity. When they actually in the beginning, early 1990s, they started blood donor screening program because we cooperate with Michael Houghton. We have the first generation anti-CV, the C100-3 for the blood donor screening. So then we can reduce the post-transfusion hepatitis C at a time. And then interferon come, although the effectiveness is very poor, only 6%. But the National Taiwan University Hospital, we conducted a clinical trial. We combined the interferon and also ribavirin, and we increased the sustained response rate or the cure rate from 6% to 42%. So this may be in 1996, the paper published. And until 2013, at the time, we have the first generation of DAA, direct antivirus, because the contribution of these 3 Nobel Laureates. So in the United States, they launched the DAA, and the government know, okay, this drug is very effective. The SVR rate can be up to 90% or even higher, 95%. So our health insurance program are trying to adopt these drugs into our system. So from 1960, the government reimbursed the pan-genotypic DAA into Taiwan. So from that time until now, I think the HCV patients is estimated to be 200,000 in Taiwan. So until 2025, I think we already treated nearly 200,000. So almost 95% of the SCD patients who was diagnosed has been treated with the SVR rate of 98%. So that's the reason why the government claims we have eliminated SCD according to the WHO target. The WHO target is we identify 90% of SCD patients and treat 80% of AJO patients and 65% reduction in SCC. But I think for the first two goals, we have already reached. But for the reduction of SCD mortality, it takes time because we have a lot of backlog patients already, although they have been treated with DA without virus, but they still have some fibrosis or cirrhosis, and liver cancer can still occur. So I think in the future, we need to have more vigorous liver cancer screening program in these patients, not only for SCD, but also for HPV cases. And nowadays, the muscle D, the liver disease, the fatty liver disease is also increasing. So they are also our target populations. I think with this achievement, the government claims that we have SCD elimination in Taiwan. I will give you that special issue later. Thank you so much.
So, according to what you just said, I think it's very difficult for other countries to replicate the success of elimination of HCV, right? Because it takes the government, the scientists, and the policy.
Yes, I fully agree with you. Actually, there are only 3 countries, except Taiwan, already achieved the WHO target. The first one is Egypt, as I mentioned in Professor Rice's slide. The other one is Georgia. And the other one, maybe the Japan is already may reach these targets, and then followed by Taiwan. So for the United States, you know, the system is quite different. And as I know, the number of new infections is outnumbered the number treated. So it's very difficult to have a reduction of SCV infected cases.
So, Charlie, please include Taiwan in your chart. And it's very important. It's the most successful example. Thank you.
Thank you.
We have the questions from upstairs. Dean of College of Management, Professor Liao.
Can you hear me?
Yes.
We are here.
Yeah, we can hear you.
Actually, a question raised by Professor Colin Butler. Professor.
Thank you for this chance. Thanks so much, Professor Rice, for your talk. In one of your answers, you briefly mentioned that artificial intelligence has capacity for harm and also good. And my question is, do you think that modern virology has any capacity for harm? And related to that, do you have any comments about the ethics of virologists, and do you see any change over time? Thank you.
We're sorry, can you repeat the question again because it's not really clear downstairs. We can't hear you because check the microphone, please.
Can you hear me better now? Sorry about this.
Yeah, I can hear you. It's just the fuzziness of it's a little bit difficult to exactly understand what you're saying.
I'll try to be brief. Thank you firstly for your talk. You commented in your answer about AI, I think, that it can do harm as well as good. Do you think modern virology can do harm? And relatedly, Do you have any comments on the ethics of some virologists, and is it changing? I hope you can understand me this time. Thank you.
Charlie, I think I can help. Charlie, I think I can help. I think you talked about this earlier in the faculty discussion session. I think his question is modern virology. If we study or we design virus by AI, for example, okay, would modern virology research do any harm to, in terms of ethics, to the human society? I think you had that question earlier.
So this was a worry about sort of the implication of AI and technologies that we have to sort of create things that are sort of nefarious in terms of, well, I don't know, it's always a concern. This is not an area that was neglected in terms of the origin of SARS-CoV-2. Although I think we feel that the virologists are not smart enough to come up with a virus that is as well adapted to spreading in the human population as that one. I think, it is, in terms of actually sort of being able to use AI in positive ways and negative ways, it's kind of like almost every technology that comes along. And I do worry, particularly seeing about some of the conflicts that are going on in the world right now, that we do have to be concerned about not just sort of disease agents that sort of pop out of the environment and affect people, but sort of back to the old sort of Cold War sort of days of sort of biosafety and bioterrorism and so on and so forth. So I think that's, in the U.S., this doesn't seem to be a topic that's been discussed a lot. And, people can do sort of a spectrum of good, bad, and ugly. And so I think it is a concern. Whether or not AI is going to sort of enhance, sort of the sort of ability for people to design things that are, sort of more pathogenic or whatever. I don't know. We already know enough about ones that are pathogenic that we don't have, really good solutions for, sort of like influenza that we're about. So I don't know. I think it is I often wonder in the United States if sort of one of the things that have not been brought up enough in terms of our discussion about pandemic preparedness and how we're in a much worse position now today than we were, when SARS-CoV-2 showed up. That maybe we need to talk more about sort of biodefense again. Maybe that will sort of get the attention of our Department of War.
Maybe I should ask a question. Do you use any AI-related technology to conduct experiments in your lab now? Use any AI technology in your lab now?
Well, I don't know. Most people don't confess. Gosh, that paragraph looks awfully good. Well, I think people do use AI to sort of ask questions and sort of see it. It seems to be becoming increasingly pervasive. I actually haven't figured out how to shut it off on my phone. Now my sort of emails show up with sort of an AI analysis that sort of precedes them. I don't know where that came from. I wasn't asking for it. No, I think it's going to be, it's here. It's going to be increasingly used. if it can, sort of help with organizing things and something, a lot of, potential time-saving, sort of benefits of AI, just in terms of practical things. So I'm not opposed to it. I think you just have, like with any science, I think you have to be your own worst skeptic. That's true more so now, perhaps, than it was in the past, just because there's so much information and misinformation, sort of flying around. So, do I use it? I don't think we have sort of a big AI presence, but it's certainly something that is being sort of discussed, almost on a daily basis. But we haven't really sort of employed it. Maybe we need to strike up some collaborations with the folks here to, sort of move more into the AI space.
Okay, there will be the last question, because the time is already up. Okay, thank you for all your questions, and thank you, Professor Rice, and thank you, Dr. Gao and Dr. Tsing. And one more question. Okay, one more question. Since President Liao request for that.
Thank you very much, President, to say the last question for me. Thank you very much for, I have a question for Professor Rice, and thank you for your inspiring talk. Our lab was the first to identify the sfRNA in Japanese encephalitis virus. Although JV is vaccine preventable, any important question in basic biology remain. My question is, how would you frame the importance of this kind of research in today's funding environment? Thank you. My question is, how did you suggest framing the importance of the basic biology research in today's funding environment? How can we get funding? Because this virus don't kill people. It's difficult to get funding. So my question is, I want to know, would you have any good suggestions to get funded easily and frame the importance of basic biology? Thank you.
Okay, I'm not sure I'm going to give away any of those secrets. Well, I think, sort of funding virology and sort of funding infectious disease is something that is certainly on my mind. Sort of at Rockefeller, sort of about a third of our, not just for infectious disease, but in terms of institutions, about a third of it is philanthropy, a third of it sort of comes from endowment income, and then a third of it, comes from the NIH. And, it's not like there's a huge surplus in the infectious disease space. And so I think that you bring up a really important area, which is important not only in funding, sort of more basic, fundamental, sort of virus and microbiology research, but also just how we craft a system where we can go from sort of virus discovery to really things that are going to help people and overcoming some of those roadblocks. So I think, part of the answer to that, sort of comes from, the government and the sort of organization of the country and supporting that kind of work. But it is complicated in the sense that there are so many steps from, sort of really sort of basic curiosity-driven research to recognizing that something that you're doing, has the potential to make a difference, and trying to sort of move that through further studies and preclinical development, and then how do you go from that to actually doing human studies and actually showing that your idea sort of works. And I think I would love to see more of a global effort to tackle some of these problems, because I think that's ultimately what's needed. But it's obviously something that, sort of we're not seeing great movement in that direction, at least in the United States today. But I think we need more cooperation. I'm excited by a new Institute for Global Infectious Disease Research that we just started at Rockefeller a few years ago. And part of the mission of that is to look for other sources of funding, but also just try and get more groups collaborating in different countries. And certainly the reach of what Taiwan has, the foundation has in all of these different sort of countries where help has been provided is a resource for infectious disease going forward. But in terms of any sort of magical funding, I guess one of the things that I didn't really completely appreciate is, sort of how much money in the United States was sort of being devoted to sort of research. You don't have that anymore, or it's sort of under threat. It really does come home. But I think that, the public needs to be educated on the importance of research, and part of that, sort of has to do with, you would think, everybody would recognize the importance of, getting a measles vaccine, or whatever. But, that's not necessarily true. And I am a little, in terms of trying to get the public more engaged, I think in some places it comes down to really sort of fundamental education. And that's why I was, sort of happy to see, sort of high school students and sort of other undergraduates here in the audience, because I think, even people in sort of early school, can ask questions and sort of recognize what's right and wrong, what's true and not true. And we need to start that earlier, but it's not easy to change the world.
Since funding is always a big issue for all the scientists. Okay, thank you, and Professor Gao, Dr. Gao and Dr. Tsing, please return to your seats. And Professor Rest, please remain on the stage. At this time, we would like to invite President of Tzu Chi University to come forward to the stage. President Liao will be presenting the special gift as a token of our gratitude and our friendship.